Chapter 3, Cells. Cell is the structural and functional unit of all living matter. What is cytology? Cytology is study of cellular structure and function. Cells vary considerably in size, shape, and function. A red blood cell, or RBC, for example, is tiny, whereas a single nerve cell may measure 4 feet in length. The shapes and structures of the cells are also very different. The RBC is shaped like a frisbee and is able to bend. The shape allows it to squeeze through tiny blood vessels and deliver oxygen and other nutrients throughout the body. Some nerve cells are very long, and many resemble bushes or trees. Their shapes enable them to conduct electrical signals quickly over long distances. Cell structure and function are closely related. Typical cell. Despite the differences, cells have many similarities. Each specialized cell, such as a nerve cell, possesses some or all of the properties of the typical cell. Cell membrane. The cell is encased by a cell membrane, also called the plasma membrane. The cell membrane separates intracellular, or intracellular means inside the cell. The cell membrane separates intracellular material from extracellular or the outside the cell material. In addition to physically holding the cell together, the cell membrane performs other important functions. One of its chief functions is the selection of substances allowed to enter or leave the cell. Because the membrane chooses the substances allowed to cross it, the membrane is said to be selectively permeable or semi-permeable. What makes up a cell membrane? The cell membrane is composed primarily of phospholipids and protein, as well as a small amount of carbohydrates. The phospholipids are arranged in 2 layers. The protein molecules in the membrane perform several important functions. They provide structural support for the membrane, act as binding sites for hormones, and poke holes or pores through the lipid membrane. These pores form channels through which water and dissolved substances flow. Substances move across the semi-permeable membrane in 2 ways. They can dissolve and the lipid portion of the membrane, as do oxygen and carbon dioxide, or lipid-soluble substances. Substances can also cross the membrane by flowing through the pores. Water and electrically charged substances such as sodium and chloride cannot penetrate the lipid membrane and must use the pores. These are called water-soluble substances. The size of the pores also helps select which substances cross the membrane. Substances larger than the pores cannot cross the membrane, whereas smaller substances such as sodium and chloride flow through easily. The solubility characteristics of the membrane also play an important role in pharmacology. Drugs are classified as lipid or fat soluble or water soluble. Drug solubility determines its distribution throughout the body. Inside the cell. The inside of the cell is divided into two compartments, the nucleus and the cytoplasm. The inside of the cell resembles the inside of a raw egg. The yellow yolk is the nucleus, and the white is the cytoplasm. Nucleus. The nucleus is the control center of the cell. In particular, the nucleus contains the genetic information and controls all protein synthesis. Most adult cells have one nucleus. Only mature RBCs have no nucleus. Surrounding the nucleus is a double-layered nuclear membrane. The nuclear membrane contains large pores that allow the free movement of certain substances between the nucleus and cytoplasm. The nucleus is filled with a fluid substance called nucleoplasm. Within the nucleoplasm are two other structures, the nucleolus and chromatin. The nucleolus, or little nucleus, synthesizes ribosomes that move through nuclear pores into the cytoplasm, where they play a role in protein synthesis. The nucleolus produces a nucleotide necessary for protein synthesis. Chromatin is composed mainly of strands of DNA, or deoxyribonucleic acid, the carriers of the genetic code. In non-dividing cells, chromatin appears as a tangled array of fine filaments. In dividing cells, however, chromatin strands coil tightly, forming DNA-containing structures called chromosomes. Cytoplasm. Cytoplasmic gel. The cytoplasm, or the gel in a cell, is found inside the cell. But outside the nucleus, like the white of a raw egg, the cytoplasm contains the cytosol, and organelles. The cytosol is the extracellular fluid and composed primarily of water, electrolytes, proteins, and nutrients. The cytosol also contains inclusion bodies, insoluble materials such as glycogen, granules, and also pigments such as melanin. The organelles, or it's called little organs, are dispersed throughout the cytoplasm. Each organelle has a specific role, next will be cytoplasmic organelles. Mitochondria. The mitochondria are tiny and slipper-shaped organelles. The number of mitochondria per cell varies, depending on the metabolic activity of the cell, how hard the cell works. The more metabolically active the cell, the greater the number of mitochondria. The liver, for example, is very active and therefore has many mitochondria per cell. Bone cells are less active metabolically and have fewer mitochondria. The mitochondrial membrane has two layers, which is the outer layer is smooth, whereas the inner layer has many folds, referred to as cristae. The enzymes associated with ATP production are located along the cristae because the mitochondria produce most of energy in the body. They are referred to as the power plants of the cell. Next is the ribosomes. Ribosomes are cytoplasmic organelles involved in protein synthesis. Some ribosomes are attached to the endoplasmic reticulum and are called fixed ribosomes. Fixed ribosomes are largely concerned with the synthesis of exportable protein, that is, protein secreted by the cell for use elsewhere in the body. Other ribosomes, called free ribosomes, float freely within the cytoplasm and generally synthesize proteins that are used within the cell. Endoplasmic Reticulum, or ER, is a network of membranes within the cytoplasm. These long folded membranes form channels through which substances, especially newly synthesized protein, move. The two types of endoplasmic reticulum that include the type containing ribosomes along its surface. It is called rough endoplasmic reticulum, or RER, because of its rough, sandpaper-like appearance. The RER is primarily concerned with protein synthesis. Protein synthesized along the RER is transported through the channels and delivered to the Golgi Apparatus for further processing. The ER that does not contain ribosomes on its surface appears smooth. It is called smooth endoplasmic reticulum, or SER. SER is primarily involved in the synthesis of lipids, steroids, glycerides, and glycogen in skeletal muscle and liver cells. Next is Golgi Apparatus. It's a series of Latin membranous sacs, proteins synthesized along the RER or called the Endoplasmic Reticulum, is a series are transported to the Golgi Apparatus through Golgi channels formed by the ER. The Golgi Apparatus puts the finishing touches on the protein. For example, a glucose molecule may be attached to a protein within the Golgi Apparatus. Golgi Apparatus is also known as the Golgi complex, Golgi body, or simply the Golgi. Is an organelle found in most eukaryotic cells. It was identified in 1897 by the Italian scientist Camillo Golgi and named after him in 1898. Golgi Apparatus is a series of Latin membrane sacs, protein synthesized along the RER, or it's called the Endoplasmic Reticulum, are transported to the Golgi Apparatus through channels formed by the ER. The Golgi Apparatus puts the finishing touches on the protein. For example, a glucose molecule may be attached to a protein within the Golgi Apparatus. A segment of the Golgi membrane then wraps itself around the protein and pinches itself off to form a secretory vesicle. In this way, the Golgi Apparatus packages the protein. Note that many of the organelles, particularly the ribosomes, ER, and Golgi apparatus, are involved in protein synthesis. Lysosomes are membranous sacs containing powerful enzymes. Lysosomal enzymes break down intracellular waste and debris, including damaged organelles, and thus help clean house. Lysosomal enzymes perform several other functions. They participate in the destruction of ingested bacteria, a process called phagocytosis. Lysosomes are also break down the contractile proteins of inactive muscles, as occurs in retired athletes and chronically bedridden persons. Cytoskeleton. The cytoskeleton is composed of thread-like structures called microfilaments and microtubules. The cytoskeleton helps maintain the shape of the cell and assess the cell in various forms of cellular movement. Cellular movement is particularly evident in muscles, which contain large numbers of microfilaments. Microtubules are the primary component of the cytoskeleton. In addition to making the cell strong and rigid, the microtubules anchor the position of the organelles within the cytoplasm. Microtubules also play a key role in cell division. They form the spindle apparatus that helps distribute the chromosomes to opposite ends of the dividing cell. Centrioles. Centrioles are paired, rod-shaped, and short microtubular structures that form the spindle apparatus in a dividing cell. Cells that have no centrioles are incapable of cell division. These include neurons, mature RBCs, skeletal muscle cells, and cardiac muscle cells. The cytoplasm surrounding the centrioles is called the centrosome. Microtubules of the cytoskeleton begin at the centrosome and spread throughout the cytoplasm. On the cell membrane, microvilli. For cells that are particularly involved with the movement of large amount of water and its dissolved solutes, the membrane forms accordion-like folds called microvilli. For singular, microvillus. The folding of the cell membrane increases surface area, thereby increasing the amount of fluid absorbed. For example, some of the cells in the digestive tract have millions of foldings, called microvilli, to absorb water and the end products of digested food. Cilia are short, hair-like projections on the outer surface of the cell membrane. Cilia use wave-like motions to move substances across the surface of the cell. For example, cilia are abundant on the cells that line the respiratory passages. The cilia help move mucus and trapped dust and dirt toward the throat, away from the lungs. Once in the throat, mucus can be removed by coughing or swallowing. The cilia therefore help clear the respiratory passages. Cigarette smoking damages the cilia and thus deprives the smoker of this benefit. Flagella, meaning whip-like, which is similar to cilia in that both are hair-like projections of the cell membrane. Flagella, however, are thicker, longer, and fewer in number. They help move the cell. The tail of the sperm is an example of a flagellum. The tails enables the sperm to swim. Movement across the cell membrane. Cells are bathed in an extracellular fluid that is rich in nutrients such as oxygen, glucose, and amino acids. These nutrients are needed in the cell and must therefore be able to cross the cell membrane. The cell's waste, which accumulates within the cell, must also be able to cross the cell membrane. Wastes are eventually eliminated from the body. A number of mechanisms assist in the movement of water and dissolved substances across the cell membrane. The transport mechanisms can be divided into two groups: passive transport and active transport mechanisms. In So there are transport mechanisms, which is called, which part of the passive is called diffusion. It's a movement of a substance from an area of high concentration to an area of low concentration. Facilitated diffusion. Helper molecule within the membrane assists movement of substances from an area of high concentration to area of low concentration. Osmosis. Diffusion of water or solvent from an area with more water to an area with less water. The water compartments are separated by semi-permeable membrane. Filtration. The pushing of water and dissolved substances from an area of high pressure to an area of low pressure. The water and dissolved substances are pushed. Active transport mechanism. Active transport pump moves a substance uphill or it's called from an area of low concentration to an area of high concentration, and requires an input of energy, ATP, or it's called the adenosine triphosphate, or ATP. Endocytosis, taking in or ingestion of substances by the cell membrane. Phagocytosis. It is engulfing of solid particles by the cell membrane. It is a cellular eating. Phagocytosis. Pinocytosis. Engulfing of liquid droplets, which is cellular drinking. Exocytosis. Secretion of cellular products. For example, protein, debris. This is secretion of cellular products out of the cell. Exocytosis. Those are the active mechanisms. And the passive transport mechanisms require no additional energy in the form of ATP or adenosine triphosphate. Passive transport is something like the downward movement of a ball. The ball is at the top of the hill. Once early released, the ball rolls downhill. The ball does not need to be pushed. It moves passively, without any input of energy. Passive transport mechanisms cause water and dissolved substances to move without additional energy, like a ball rolling downhill. Active transport mechanisms require an input of energy in the form of ATP. Active transport is like the upward movement of a ball. For the ball to move uphill, it must be pushed, therefore requiring an input of energy.